Hello. Most of the slides in this mini lecture come from research seminars that I have given recently or plan to give. The topic is still changes in the brain with chronic nicotine. Curiously enough, in 1983 it was first noticed that in an animal given chronic nicotine maintained or repeated for a week or two, there were more nicotinic receptors, that is, they were upregulated. This is curious because most of us would have thought that during drug addiction, a component of tolerance would be actual decreases in receptors. How this played out and what causes it, I will tell you in this lecture. So we are still trying to go all the way from genes to behavior, or from behavior back to genes. And as stated, chronic nicotine increases the number of nicotinic receptors. These are functional nicotinic receptors. But interestingly enough, this so-called upregulation actually displays remarkable selectivity at every level that has been thus far examined. So the selectivity occurs only in some brain regions, only in some cell types, dopaminergic cell types versus cell types that make GABA or those that make glutamate. The upregulation occurs only in some subcellular compartments, axon terminals versus somatodendritic. And only some subtypes of nicotinic receptors are upregulated, mostly high sensitivity to nicotine rather than low sensitivity to nicotine. And furthermore, there may even be a dependence on the detailed stoichiometry of the nicotinic receptors. So let's follow this story. This is a quick summary, which you do not need to remember in any way for quizzes or exams. Here are diagrams of key regions of the brain, the VTA and the striatum, the medial habenula, which you've heard in a previous talk, and the hippocampus, which you've heard about in previous talks. This table basically tells us where, after we've given chronic nicotine for a week or two, cells grow new nicotinic receptors. In some cells, there are no new nicotinic receptors at all. In others, there are new nicotinic receptors on the terminals, projecting to another area, but not on the cell body. In others, there are nicotinic receptors both on the cell body and on the terminals. Now, one point to be made is that, as you can see, upregulation is quite robust on GABAergic cells. These are cells that usually inhibit other cells. And so the paradox that I stated, how could upregulation be associated with tolerance, involves a change in sign. It involves the fact that we have nicotinic receptors upregulated, but they are in inhibitory neurons, not on excitatory neurons. Let's go on now and talk about another aspect of upregulation. And this is the connection with desensitization. You'll remember that when we learned about nicotinic receptors, we learned that they can be activated and then they can become desensitized. And I told you that there's presently a very detailed discussion about which action of nicotine causes activation and which causes desensitization. Either activation or desensitization can be amplified by upregulation. The change is greater when it gets turned on or when it gets turned off. But now let's ask the question, how does this upregulation occur? Most biologists would say, oh, upregulation, that means we are activating new genes, making new nicotinic receptors. But that is apparently not what's happening in this case. Let's go back and remember that nicotine in the lungs can be a neutral molecule in that in its neutral form, it passes through 6 plasma membranes in about 20 seconds. So this is an event that takes place about 150 billion times a day when a smoker takes a puff. We get neutral nicotine getting into the brain. And as usual, it can gain or lose a proton, again, become charged, in just milliseconds. Let's follow what happens when nicotine becomes extracellular in the brain. Well, according to the classical concepts that I have been teaching you, when nicotine is in the brain and is charged, it goes through the usual pathway, finds nicotinic receptors on the surface of a cell, activates those nicotinic receptors, and according to the outside-in mechanism, those receptors open, calcium flows in, and we have the various second messenger pathways activated that I've told you about in the previous lecture. And most neuroscientists are comfortable with the idea that this is the basis for the actions of long-term nicotine. Now comes the new fun. We're going to get rid of that classical pathway entirely and talk about inside-out actions, which lead to the upregulation of a special subclass, the alpha-4 beta-2 nicotinic receptor. Again, nicotine is extracellular. Again, it can gain or lose a proton. And this is roughly half and half, with and without protons. Now, it has been known for 50 years that nicotine can also permeate nerve cell membranes, gets right into the cell. And in nerve cell membranes, papers in 2005, excellent, very important papers from other labs, showed that nicotine can also enter the endoplasmic reticulum. I have to tell you that of the 100 or so neuroscientists I've talked with about inside-out mechanisms over the last 5 years, all but one or two have said, "Oh, I did not know that nicotine enters neurons." Don't be surprised if you did not know this. Now, inside the endoplasmic reticulum, a remarkable series of events occurs. Nicotine can again gain a proton, and in its protonated form, it can bind to nicotinic receptors which are being made in the endoplasmic reticulum. That's where most membrane proteins get made. Now, alpha-4 beta-2 nicotinic receptors have the curious characteristic that many of them get trapped for long periods of time in the endoplasmic reticulum. This happens in real neurons, it happens in cultured cells, it happens in the brain. We don't know its selective advantage, but it does happen. Nicotine, as you'll remember, binds at the interface between two subunits, and this helps the subunits assemble. The assembled nicotinic receptors are more able to leave the endoplasmic reticulum. Now, if you've taken cell biology in the last 10 years, you'll know that this occurs via the so-called COP2 vesicle, which contains 5 key proteins. The important part is that assembled receptors have some sequences hidden that enable them to exit the endoplasmic reticulum more readily. And then the usual secretory pathway takes over. The Golgi complex, the early endosome, and secretion pathways, and there is some backing and forthing as well. The end result is that this increased flux of nicotinic receptors from the endoplasmic reticulum caused by the action of nicotine leads to more nicotinic receptors on the cell membrane, on the plasma membrane, and that is what we call chaperoning, and that's what leads to upregulation. So in the endoplasmic reticulum, the nicotine becomes a chaperone for nascent nicotinic receptors. So, somewhat surprisingly, the upregulation of nicotinic receptors, which seems to be so key for changed circuits in nicotine addiction, does not come from activating genes. It comes from a so-called post-translational process. The various colors on these vesicles are a pH code, and I won't discuss it Let me give you some more feeling for the importance of the upregulation process. First of all, the EC50 for upregulation, the half maximal nicotine concentration, is around 37 nanomolar, and this is 10 times smaller than the peak nicotine concentration during smoking, and it's also too small to activate any appreciable number of nicotinic receptors. And so, in fact, chaperoning, leaving the endoplasmic reticulum, is actually occurring at nicotine concentrations that are lower than those that activate surface receptors, which is why we think it is so important in a person who has smoked recently and still has some nicotine in his organelles. The physical chemistry of chaperoning is not yet certain, but essentially you may remember this diagram where if a ligand and receptor interact with each other, examine each other long enough, they can find states of very high affinity. And apparently those states of very high affinity are protected against destruction in the endoplasmic reticulum or in the cytoplasm. There's another effect. As you'll remember, nicotine binds at the interface between subunits, and therefore, by most thermodynamic arguments, it must stabilize these interfaces, and therefore nicotine helps to stabilize the assembled receptor of 5 subunits. Now, my friends say that this is not chaperoning, it's actually matchmaking to extend the social commentary. So these aspects of chaperoning and matchmaking are what help nicotine to upregulate nicotinic receptors. Let me show you a movie that demonstrates this process. It's actually 2 movies. Good. On the left is a cell in tissue culture, a neuron-like cell, neuroblastoma, that has been modified to express fluorescent alpha-4 subunits fused to GFP with non-fluorescent beta-2 subunits. And the special microscope that's used here is called total internal reflection fluorescence microscopy. So, in the cell that has been sitting for 48 hours with no drugs, the pattern, whoops, let's try that again, there we are. In the cell that has been sitting there for 48 hours, the pattern looks like not much movement, and it looks reticular. In fact, the nicotinic receptors seem to be stuck by and large in the endoplasmic reticulum. Here on the right, on the other hand, this is a cell that has been incubated in nicotine for 48 hours. A rather low concentration of nicotine, lower than would activate receptors, and you can see that the pattern is quite different. There are receptors moving around inside the cell along tracks. There are large blobs of receptors. These have all left the endoplasmic reticulum. And furthermore, you can see that there is a diffuse haze all around the cell that's actually on the plasma membrane, and that this diffuse haze is brighter in the cell that's been exposed to nicotine than in the cell that has not been exposed to nicotine. That is, of course, the upregulation on the plasma membrane itself, the result of the chaperoning and the receptors that have left the endoplasmic reticulum and have appeared on the plasma membrane. Summarizing, changes in the brain during chronic exposure to nicotine. In a modern hypothesis, selective upregulation of nicotinic receptors is actually necessary and sufficient for the early days of nicotine dependence, hours, days, and weeks. This selective upregulation occurs because of chaperoning. It occurs because receptors are sometimes unable to leave the endoplasmic reticulum and can do so more easily when nicotine is present. And the changes in the functions of circuits are what change the function of the nervous system. And so one could say that selective upregulation instantiates some phenomena that are typically invoked to explain the neuroscience of drug abuse. These phenomena include such words as adaptation, neuroadaptation, plasticity, compensation, homeostasis. These words are actually not mechanisms themselves. They're more like adjectives. We can think about homeostatic mechanisms and about plastic mechanisms and about adaptive mechanisms. And the detailed molecular changes that I've told you about in this lecture seem to instantiate those mechanisms. Now, we certainly do not understand all of nicotine addiction, and several processes are not well understood. These include the somatic signs of withdrawal and also the stress-induced use and relapse of nicotine. For instance, the person who has not smoked in a year or so gets into a car accident, gets offered a cigarette, and begins to smoke again. We do not know how this occurs. So there are many, many lessons to be learned, but you can see how scientists are now beginning to tease apart the events that occur inside a neuron and in neuronal circuits that have been exposed chronically to nicotine.